Regulation of glycolysis and gluconeogenesis. Molecular biochemistry I. Gluconeogenesis. Regulation of glycolysis and gluconeogenesis. Copyright 1999 to 2007 by Joyce J. Devon. All rights reserved. Gluconeogenesis occurs mainly in liver. Gluconeogenesis occurs to a more limited extent in kidney and small intestine under some conditions. Synthesis of glucose from pyruvate utilizes many of the same enzymes as glycolysis. 3 glycolysis reactions have such a large negative DG that they are essentially irreversible. Hexokinase, or glucokinase. Phosphofructokinase. Pyruvate kinase. These steps must be bypassed in gluconeogenesis. 2 of the bypass reactions involve simple hydrolysis reactions. Hexokinase or glucokinase, glycolysis, catalyzes. Glucose plus ATP, glucose 6-phosphate plus ATP. Glucose 6-phosphatase, gluconeogenesis, catalyzes. Glucose 6-phosphate plus H2O, glucose plus pi. Glucose 6-phosphatase enzyme is embedded in the endoplasmic reticulum, membrane in liver cells. The catalytic site is found to be exposed to the lumen. Another subunit may function as a translocase, providing access of substrate to the active site. Phosphofructokinase, glycolysis, catalyzes. Fructose 6P plus ATP, fructose 1,6 bis plus ATP. Fructose 1,6 bisphosphatase, gluconeogenesis, catalyzes. Fructose 1,6 bis plus H2O, fructose 6P plus pi. Bypass of pyruvate kinase. Pyruvate kinase, last step of glycolysis, catalyzes. Phosphenolpyruvate plus ADP, pyruvate plus ATP. For bypass of the pyruvate kinase reaction, cleavage of 2 p bonds is required. DG for cleavage of 1 p bond of ATP is insufficient to drive synthesis of phosphenolpyruvate, PEP. PEP has a higher negative DG of phosphate hydrolysis than ATP. Bypass of pyruvate kinase, 2 enzymes. Pyruvate carboxylase, gluconeogenesis, catalyzes. Pyruvate plus HCO3 plus ATP, oxaloacetate plus ADP plus pi. PEP carboxygenase, gluconeogenesis, catalyzes. Oxaloacetate plus GTP, PEP plus GDP plus CO2. Contributing to spontaneity of the two-step process. Free energy of 1 p bond of ADP is conserved in the carboxylation reaction. Spontaneous decarboxylation contributes to spontaneity of the second reaction. Cleavage of a second p bond of GTP also contributes to driving synthesis of PEP. Pyruvate carboxylase uses biotin as prosthetic group. Biotin has a 5C side chain whose terminal carboxyl is an amide linkage to the E amino group of an enzyme lysine. The biotin and lysine side chains form a long swinging arm that allows the biotin ring to swing back and forth between 2 active sites. Biotin carboxylation is catalyzed at one active site of pyruvate carboxylase. ATP reacts with HCO3 to yield carboxyphosphate. The carboxyl is transferred from this P intermediate to N of a urido group of the biotin ring. Overall, biotin plus ATP plus HCO3 carboxybiotin plus ADP plus pi. Carboxybiotin plus pyruvate. At the other active site of pyruvate carboxylase, the activated CO2 is transferred from biotin to pyruvate. Carboxybiotin plus pyruvate. Biotin plus oxaloacetate. View in animation. Pyruvate carboxylase, pyruvate oxaloactate, is allosterically activated by acetyl-CoA. Oxaloacetate tends to be limiting for Krebs cycle. When gluconeogenesis is active in liver, oxaloacetate is diverted to form glucose. Oxaloacetate depletion hinders acetyl-CoA entry into Krebs cycle. The increase in acetyl-CoA activates pyruvate carboxylase to make oxaloacetate. Avidin, a protein in egg whites with a B barrel structure, tightly binds biotin. Excess consumption of raw eggs can cause nutritional deficiency of biotin. The strong avidin to biotin affinity is used by biochemists as a specific glue. If it is desired to bind two proteins together for an experiment, biotin may be covalently linked to one protein and avidin to the other. Explore with CHIME the biotinyl domain of a carboxylase and the avidin-biotin complex. PEP carboxykinase catalyzes GTP-dependent oxaloacetate PEP. PEP carboxykinase catalyzes GTP-dependent oxaloacetate PEP. It is thought to proceed in two steps. Oxaloacetate is first decarboxylated to yield a pyruvitinylate anion intermediate. Phosphate transfer from GTP then yields phosphenolpyruvate, PEP. In the bacterial enzyme, ATP is pi donor instead of GTP. In this crystal structure of an E. coli PEP carboxykinase, pyruvid is at the active site as an analog of PEP/oxaloacetate. A metal ion such as Mn++ is required for the PEP carboxykinase reaction, in addition to a Mg++ ion that binds with the nucleotide substrate at the active site. Mn++ is thought to promote pi transfer by interacting simultaneously with the enolate oxygen atom and an oxygen atom of the terminal phosphate of GTP or ATP. The source of pyruvid oxaloacetate for gluconeogenesis during fasting or carbohydrate starvation is mainly amino acid catabolism. Some amino acids are catabolized to pyruvid, oxaloacetate, or precursors of these. Muscle proteins may break down to supply amino acids. These are transported to liver where they are deaminated and converted to gluconeogenesis inputs. Glycerol, derived from hydrolysis of triacylglycerols in fat cells, is also a significant input to gluconeogenesis. Summary of gluconeogenesis pathway. Gluconeogenesis enzyme names in red. Glycolysis enzyme names in blue. Glycolysis and gluconeogenesis are both spontaneous. If both pathways were simultaneously active in a cell, it would constitute a futile cycle that would waste energy. Glycolysis. Glucose plus 2 Namibian dollars plus 2 ADP plus 2 pi. 2 pyruvid plus 2 NOD plus 2 ATP. Gluconeogenesis. 2 pyruvid plus 2 NOD plus 4 ATP plus 2 GTP. Glucose plus 2 Namibian dollars plus 4 ADP plus 2 GDP plus 6 pi. Questions. 1. Glycolysis yields how many P? 2. Gluconeogenesis expends how many P? 3. A futile cycle of both pathways would waste how many? P per cycle? 2. 6. 4. To prevent the waste of a futile cycle, glycolysis and gluconeogenesis are reciprocally regulated. Local control includes reciprocal allosteric regulation by adenine nucleotides. Phosphofructokinase, glycolysis, is inhibited by ADP and stimulated by AMP. Fructose 1,6-bisphosphatase, gluconeogenesis, is inhibited by AMP. The opposite effects of adenine nucleotides on phosphofructokinase, glycolysis. Fructose 1,6-bisphosphatase, gluconeogenesis, ensures that when cellular ATP is high, AMP would then be low, glucose is not degraded to make ADP. When ATP is high, it is more useful to the cell to store glucose as glycogen. When ATP is low, AMP would then be high, the cell does not expend energy in synthesizing glucose. Global control in liver cells includes reciprocal effects of a cyclic AMP cascade, triggered by the hormone glucagon when blood glucose is low. Phosphorylation of enzymes and regulatory proteins in liver by protein kinase A, CAMP-dependent protein kinase, results in inhibition of glycolysis, stimulation of gluconeogenesis, making glucose available for release to the blood. Enzymes relevant to these pathways that are phosphorylated by protein kinase include pyruvate kinase, a glycolysis enzyme that is inhibited when phosphorylated. CREB, CAMP response element binding protein, which activates, through other factors, transcription of the gene for PEP carboxygenase, leading to increased gluconeogenesis. A bifunctional enzyme that makes and degrades an allosteric regulator, fructose 2,6-bisphosphate. Reciprocal regulation by fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate stimulates glycolysis. Fructose 2,6-bisphosphate allosterically activates the glycolysis enzyme phosphofructokinase. Fructose 2,6-bisphosphate also activates transcription of the gene for glucokinase, the liver variant of hexokinase that phosphorylates glucose to glucose 6-phosphate, the input to glycolysis. Fructose 2,6-bisphosphate allosterically inhibits the gluconeogenesis enzyme fructose 1,6-bisphosphatase. Recall that phosphofructokinase, the rate-limiting step of glycolysis, is allosterically inhibited by ADP. At high concentration, ATP binds at a low-affinity regulatory site, promoting the tense conformation. Sigmoidal dependence of reaction rate on fructose 6-phosphate is observed at high ATP. PFK activity in the presence of the globally controlled allosteric regulator fructose 2,6-bisphosphate is similar to that at low ATP. Fructose 2,6-bisphosphate promotes the relaxed state, activating phosphofructokinase even at high ATP. Thus activation by fructose 2,6-bisphosphate, whose concentration fluctuates in response to external hormonal signals, supersedes local control by ATP. Phosphofructokinase 2, PFK2, domain catalyzes. The allosteric regulator fructose 2,6-bisphosphate is synthesized and degraded by a bifunctional enzyme that includes two catalytic domains. Phosphofructokinase 2, PFK2, domain catalyzes. Fructose 6-phosphate plus ATP fructose 2,6-bisphosphate plus ADP. Fructose biophosphatase 2, FPase 2, domain catalyzes. Fructose 2,6-bisphosphate plus H2O fructose 6-phosphate plus pi. Bifunctional PFK2/FPase 2 assembles into a homomer. Adjacent to the PFK2 domain in each copy of the liver enzyme is a regulatory domain subject to phosphorylation by CAMP-dependent protein kinase. Which catalytic domains of the enzyme are active depends on whether the regulatory domains are phosphorylated. View in animation. CAMP-dependent phosphorylation of the bifunctional enzyme activates FPase 2 and inhibits PFK2. Fructose 2,6-bisphosphate, thus decreases in liver cells in response to a CAMP signal cascade, activated by glucagon when blood glucose is low. Downstream effects of the CAMP cascade. Glycolysis slows because fructose 2,6-bisphosphate is not available to activate phosphofructokinase. Gluconeogenesis increases because of the decreased concentration of fructose 2,6-bisphosphate, which would otherwise inhibit the gluconeogenesis enzyme fructose 1,6-bisphosphatase. Summary of effects of glucagon CAMP cascade in liver. Gluconeogenesis is stimulated. Glycolysis is inhibited. Glycogen breakdown is stimulated. Glycogen synthesis is inhibited. Free glucose is formed for release to the blood. The Cori cycle operates during exercise. For a brief burst of ATP utilization, muscle cells utilize P stored as phosphocreatine. Once phosphocreatine is exhausted, ATP is provided mainly by glycolysis, with the input coming from glycogen breakdown and from glucose uptake from the blood. Aerobic fat metabolism, discussed elsewhere, is more significant during a lengthy period of exertion such as a marathon run. Lactate produced from pyruvate passes via the blood to the liver, where it may be converted to glucose. The glucose may travel back to the muscle to fuel glycolysis. The Cori cycle costs 6p in liver for every 2p made available in muscle. The net cost is 4p. Although costly in p bonds, the Cori cycle allows the organism to accommodate to large fluctuations in energy needs of skeletal muscle between rest and exercise. The equivalent of the Cori cycle also operates during cancer. If blood vessel development does not keep pace with growth of a solid tumor, decreased O2 concentration within the tumor leads to activation of signal processes that result in a shift to anaerobic metabolism,